In this video, we are going to finish the topics in chapter 15. As I mentioned previously, ATP is the cell's energy currency. ATP stands for adenosine triphosphate. That's where the A, the T, and the P come from. So what that means is that whenever we are releasing energy inside our body, we don't release it like we see a flame in terms of a combustion. Inside of the body, we are going to be producing this biomolecule that is called ATP, and understand that one of the reasons why we conceptualize that this is how energy is released inside the body is because the hydrolysis of this molecule releases energy. So, as you can see here, in catabolic pathways that are considered oxidative, they are also considered exergonic because remember that in this process, when we take nutrients like glucose and we combine it with oxygen, then we're going to be producing CO2 and water, and we're taking the molecule of ADP, we're combining it with inorganic phosphate to make that ATP. Now, the hydrolysis of this ATP molecule, as we're going to see in some of the pathways, can be utilized for processes that are anabolic, which are considered reductive and endergonic, to take some precursor molecules into products of anabolism. Now, let's look at ATP in its structural level. As I mentioned, ATP is adenosine triphosphate. What that means is that this is a nucleotide derivative. And even though ATP, it is utilized to extend the molecule of DNA, here we have a more particular purpose for it. So as you can see, ATP is composed of adenine as a nitrogen base, ribose as a sugar, and because it is a triphosphate, it has 3 phosphate groups coming together, and these 3 phosphate groups are going to make a functional group that is called a phosphoric anhydride. Now, every time we hydrolyze the bond between those 2 phosphates, as you can see, we're going to release at least 7.3 kilocalories of energy per mole. So that's where the energy of the hydrolysis actually comes from. Now, it is important to review that because these reactions are going to be either oxidative or reductive, what do we mean by oxidation reductive reactions in metabolism? So when it comes to oxidation and reduction, we are going to relate it to concepts of general chemistry and organic chemistry. In general, understand that oxidation corresponds to the loss of electrons. But because we are talking about biomolecules that contain carbon, hydrogen, and oxygen, oxidation refers to the loss of hydrogen. It can also mean the gain of oxygen, or we can have more bonds to oxygen. Understand that when we're talking about reductions, in general, reducing a molecule is gaining electrons. But because, again, our biomolecules are composed of carbon, hydrogen, oxygen, reductions can also mean gaining hydrogens. It can mean loss of oxygen, or we have less bond to oxygen in these biomolecules. And we can see it illustrated for oxidations, specifically in the bottom. So when it comes to oxidations and reductions in metabolism, we can see here that we are going to see throughout the chapters that we're going to be discussing in the upcoming weeks how catabolism is going to be oxidative in nature. And we're going to see how energy is going to be released in the form of ATP. While, which as you can see, it is given in the left side of this figure. Anabolism, on the other hand, is going to be more of a reductive process, and we're going to be observing how energy it is required in these systems. Now, we're going to have very selective examples of each, so understand that because of that, we're going to focus on certain topics for catabolic pathways and anabolic pathways. Now, when it comes to metabolism, we also have some important biomolecules that are going to be aiding our reactions. And we can see here that these biomolecules specifically are going to be coenzymes. Okay? The ones that we are going to be focusing on in terms of the coenzymes are going to be NADH in its high energy form, which as you can see is going to be NAD+, FADH in its high energy form, the low energy form is FAD, and lastly we're going to be seeing acetyl-CoA, which is its high energy form, coenzyme ASH is going to be its low energy form. But we're going to see how these coenzymes are going to be playing a role in these reactions that are going to be either oxidative or reductive in terms of metabolism. Remember that in addition to that, ATP is going to be the energy resource that we're going to be observing in some of these reactions. We already discussed the different structures that are the important components of ATP. But just as a reminder, ATP it is composed of adenine, ribose sugar, and a triphosphate. Now remember that the hydrolysis of ATP for each one of these phosphates that can be removed is going to contribute 7.3 kilocalories per mole. Let's continue talking about the important coenzymes, starting with nicotinamide adenine dinucleotide. So when it comes to this, you can see here the structure of nicotinamide. So nicotinamide is actually composed of nicotinamide bonded to a ribose molecule, okay, that is bonded to another ribose molecule, okay, and this is adenine. Now, when it comes here, the reason why it's a dinucleotide, because as you can see, we have two of the ribose sugars connected to each other through a diphosphate, and each of them have their component, which as you can see in the top is the nicotinamide, and in the bottom is going to be that adenine. Now, the important thing to remember when it comes to this structure is what are the components of your coenzyme, and importantly, what is the reduced form. And as you can see in this reduced form, what we're doing is that we are increasing the number of hydrogen atoms bonded to the molecule, and we can observe that particularly in the nicotinamide portion of our molecule, we have increased the number of bonds to hydrogen. Another important coenzyme that we are going to see specifically once we get into the Krebs cycle, which is in chapter 19, is Flavin adenine dinucleotide. In Flavin adenine dinucleotide, as you can see, similar to NAD, this is going to be and adenine derivative. So these are nucleotide derivatives, just like ATP, but this is going to be involved in reactions for metabolism. Now, when it comes to flavin adenine dinucleotide, as you can see, is going to be composed of a flavin, ribitol, which as you can see is a carbohydrate. Then that ribitol is bonded to a diphosphate that then it is also bonded to an adenine molecule and a ribose sugar. In general, one of the things that I want to illustrate here is that when we look at the structure for it, the low energy form, which is the first one, is actually transformed into the reduced form or the high energy form of the molecule by again increasing the number of bonds to hydrogen. In this reaction, in this illustration, you can see that 2 hydrogens were incorporated into the FAD molecule to make FADH2, which is a high energy molecule. Now, the last important biomolecule is going to be coenzyme A, and you can see that coenzyme A is made by the following components coming together. It's going to have an amino ethane thiol, which is here illustrated by this green box, and I'm highlighting it in blue. It also is composed of pantothenic acid, which is going to be this portion of the molecule. And then, lastly, coenzyme A has a 3' phosphorylated ADP. So as you can see, all of the 4 different coenzymes that I mentioned, or at least, let me fix that. So when it comes to this, NAD, FAD, and coenzyme A, which are going to be involved in reactions, as you can see, contain adenine. ATP, we're going to see its utilization, but not specifically aiding reactions. And as we move throughout this chapter, we're going to see its use. So as I was mentioning, coenzyme A also has a 3' phosphorylated ADP, and we can see specifically in the molecule, it is the last component that we see all the way to the right. Now, why is coenzyme A important? Because one of the things is that we're going to be observing how coenzyme A can actually be acylated. So if we put an acetyl group on it, which I'm highlighted in green, is given in this molecule by the yellow box. That is going to actually create an important functional group that is known as a Thio Ester. This Thio Ester, which I'm going to highlight in blue, is where we have a carbonyl bonded to the sulfur. So this Thio Ester link, we are going to see that when it is utilized in reaction, it actually is going to release energy. So when our acetyl-CoA gets hydrolyzed, and I know the hydrolysis is happening because we have the addition of water to the molecule, you can see how energy is released from the hydrolysis. But we are going to look at how these molecules are utilized in reactions in the future when we continue to discuss specific pathways for metabolism.